Hi, in this video, I'll be talking about techniques to detect protein-protein interaction. I'll be talking about several techniques, but at least focusing on three major techniques which could be used to detect protein-protein interaction. First technique I'll be talking about is co-immunoprecipitation followed by Western blot or mass spec. So this process is very useful in terms of detecting protein-protein interaction in vivo. But there is a limitation. I'll be talking about that. So for this process, you need the cell lysate or the tissue homogenate, anything from which you want to detect the protein. I mean, where you want to detect the two protein of interest are interacting with each other or not. And then when the cell has been lysed in the tube, you would be adding the antibody coated magnetic beads. Now these beads would have the antibody of interest for a known protein. Let's say you want to understand whether protein A and B are interacting with each other. So these beads would have the antibodies against protein A because you know in this solution protein A is present. You want to detect whether protein B is interacting with protein A in this solution or not. Then what would you do? You would incubate it for some time and then you would use magnetic separation to get the beads out. You would hope by that time, if protein A, you know the protein A would be there. So protein A would be pulled down by the beads. Now expectation is if protein B is interacting with protein A, along with protein A, protein B would be also pulled down. So in the lysate, we should be able to detect protein B if we pull down protein A. So from the pull down fraction of protein A, we would take the pull down fraction and load it onto the gel and run it in SDS page. Then we would perform a Western blot, transfer that gel into membrane and perform a Western blot to understand whether the protein B is present or not. And the result looks something like this. So you can see we wanted to understand whether protein A interacts with protein B or not. So we need an antibody, and the protein A antibody we have used. So we are confident that we have pulled down the protein A. That is why you can see the band corresponding to protein A, right? Then you want to use an antibody, anti B. Now this anti B is against protein B. So in this pull down fraction of protein A, if anti B is if B is present and they're interacting with each other, then you would see a band like that, and that's what you see here. You see a band. Now in the previous lane, you see you have a negative control, like you haven't given any antibody against protein A, so literally you didn't do any pull down of protein A. So even after giving antibody of protein B, you are not able to detect it, right? Because we know that only we would detect in this Western blot if we have the protein interacting with my protein of interest. Otherwise, we won't find it. So the disadvantage of this method is that all the time for all the protein you don't have the antibodies ready, and some antibodies are not very efficient in terms of pull down or detection, so there could be problem. But overall, this is a good method of understanding protein protein interaction in vitro as well as in vivo. Second, we'll talk about yeast to hybrid system, one of the best method of studying protein protein interaction in vitro. Before we start and try to understand what yeast to hybrid system is, we talk about a little bit of budding yeast metabolism. So budding yeast has gal gene, and the gal gene uh, give rise to galactose. Give rise to enzymes which break down galactose, and it is important for its metabolism. And gal4 is the protein which kind of helps the transcription of this gene. Now gal4 has two domains, activation domain and the DNA binding domain. So the DNA binding domain is important for binding to the DNA, where activation domain interacts with the polymerase and thereby initiate the transcription of the gal gene. However, if the gal gene is not transcribed, there would be metabolic defect in these yeast. So we have to clone our gene of interest in expression plasmid. So let's say, and we have to like make a fusion protein kind of thing. So here you can see one protein is now interacting with so there is our bait plasmid, and we know this protein is there, and we want to understand with this bait which other fish plasmid interact. So there are fish plasmid, and there could be multiple different fish plasmid. Then you know that if bait and fish interact with each other, only then the reporter gene underneath the promoter would be transcribed, and that is happening. And if they are not interacting with each other, then the gene transcription won't happen. Now you have a his reporter. So normally what happens is like these yeast are unable to grow without this histidine. So what happens is like if this bait and fish are interacting with each other, only then the his reporter would be expressed and the yeast would be able to grow in this medium. So then you can have a bait plasmid and multiple different fish plasmid. With the multiple different fish plasmid, you can try to understand whether a plethora of protein is interacting with your bait or not. And this is kind of the data you get from the experiment. So you have your bait plasmid and multiple fish plasmid, and in this situation you can see in these two situations highlighted in red arrows, there the bait is interacting with the fish, and that is why you can see yeast colonies there, right? In other situation you don't see yeast colonies because all of them are dead, because none of them expressed his reporter, and that is why they cannot metabolize histidine and they are not surviving. That is how we can understand whether a plethora of protein is interacting or not. So here whatever is marked in red arrow are interacting with each other, these two proteins, and other proteins are not interacting with each other. And this is a great method, but it is limited to in vitro, not in vivo. There is another good method for in vitro protein-protein interaction, which is fluorescence microscopy based. So this is called fluorescence resonance energy transfer. In order to do that, you need to have your protein A, and you suspect your protein A is interacting with protein B. So protein A should be cloned in a vector containing CFP. So there a fusion protein would be produced, and protein B would be with a vector. It would be in a vector with a YFP. So and then these vectors would be transvected into the cell line or the tissue, electroporated, anything like that. And then you check in fluorescence microscope the following things. So let's say there is no interaction between A and B. Then what would happen? You try to illuminate the CFP, which is which is fused with A, and you would try to illuminate with 434 nanometer live, and you would get a emission at 476. So none of the fret has occurred in this situation, right? So if there is no interaction, no fret. But indeed, if there is interaction, then these fret would happen, and these two proteins are close enough for the fret to happen. In that situation, if you illuminate with 434 nanometer light instead of getting a emission at 476 nanometer, you're getting a shifted to 526 nanometer. So in this situation, you can understand you illuminated CFP, but you are getting a emitted light in a YFP range. So then you can calculate quantitatively about their kinetics of interaction. So this fret is very important and useful technique for detecting protein-protein interaction as well. But the disadvantage of this method is like most of the cases it is not if the sensors are not made or a specific transgenic line is not made, it is not available in vivo situation. So it is mostly in vitro types. So and also sometimes fret efficiency is less, and sometimes the proteins are so big or present in a huge molecular complex, so then we get a false positive result. But still these three techniques are golden techniques for detecting protein-protein interaction, both in vitro and in vivo. So if you like this video, give it a quick thumbs up, and don't forget to like, share, and subscribe. Thank you.